Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event that is endorsed by the International Society for Extracellular Vesicles. So I'd like to welcome Rosalie, Martel, and David Juncker. So David, I want to hand this over to you. You're going to tell us a little bit about what your lab does, and then you can introduce Rosalie. So thanks for joining.
Thank you so much for the invitation. It's a great pleasure to be here. And I also want to actually commend the ISF Society for creating, and you, Ken, for creating this online seminar and for giving a platform to young researchers to present their work. I think that's really great. It's a great way to promote engagement and give a chance to the members to participate. And so I'll just take a few minutes to briefly introduce our lab before I hand it over to Rosalie. So we, and also actually thank the Society for giving chance to labs like ours, which is a technology development lab. And I think I'm trying to switch here my slide. So we're a technology development lab that work on a series of nanobioengineering technology, and maybe I say technology development, but actually the way I really think about it, so we come up with ideas. So we think about challenges, be it clinical or biological or technological challenges, and come up with new methods to analyze them. And over the years, we have gone in a quite a diverse direction, which is maybe linked to my interest and the fact that microfluidics and microtechnology is one of the bases, can basically serve many different applications. And so one of the latest one is extracellular vesicle analysis, which we'll talk about today a lot. But we're also looking at circulating tumor cells and maybe actually also analyzing EVs for them, organ on a chip, lab on a chip. And well, we have a lot of 3D printing, which is helping a lot of different projects, as well as microscopy, which is actually something we're also working with EVs. And so in EVs, actually today, Rosalie's paper is actually the first paper of our lab on EVs. And this has been a long story. I mean, we started actually many years ago. But like one of my students, biology students, remarked that, you know, when you do biological research, you have a hypothesis, and if it doesn't pan out, you still have some results. But when you do technology development and you want to have a new technology to measure something, if it doesn't work, then maybe there's not that much that you can publish on that. And that has been maybe a bit of our challenges. But actually right now we're really very excited because a lot of things are coming to fruition. And so I'll just give you a little bit of taste. Actually, we have just one paper that we were able to publish on arXiv yesterday, which is the work by Molly. And we're also very interested to actually get community feedback on that paper, because here we're looking at something that at the co-expression, and particularly the co-enrichment of proteins on EVs. And I think the basic premise is that, well, things can be co-expressed, but what's really important in biology is whether things are associated in a manner that goes beyond what would be stochastically expected. So they're either from biological mechanism, maybe they're associated in a way that is well above the ratio that one might express. And here's a brief illustration. Let's say we have a set of EVs, and we look at the expression of the protein recognized by this red star antibody. And we see that for 4 EVs, we have roughly 4 of these co-expression of these EVs, of these proteins. But then let's say what we can see, if you capture it with a different antibody, the co-expression might be much higher. Let's say if you have two highly abundant proteins, and when you measure them simultaneously, you might see they're not co-expressed to the way that might be expected. That would mean negative co-enrichment. They don't associate together. Or conversely, you'd have 2 proteins that have a moderate expression at the base level, but when you look then at the expression of one, you always see the expression of the other one. And that would mean in a subpopulation, they're strongly co-enriched. And we argue that this could be a very important way of uncovering biological function or association in EVs. And that's basically the exploration and arguments we build up in this arXiv paper. Another exciting work that we're having, and that's coming to fruition here, and that actually was a lot of efforts over the last few years, is single EV analyzers. There were a lot of iterations of that, but what we've developed is a way of doing single EV analyzers with a common epifluorescence microscope that we are expanding with a beam splitter. And what we can do is that using ISCAT, so interference scattering method, we can take an image before EVs are applied, which is the image with nothing. Then we apply the EVs, you see a lot dark little spots appearing. And then we can also, because it's in a flow cell, add fluorescent labels. and detect fluorescent signal, and then and evaluate the scattering signal, which is proportional to the density and the size, and the fluorescent signal simultaneously in our flow cell. And what we're seeing here from our data is that, for example, the sensitivity and specificity is outperforming what we at least could get in our hands with nanoflow cytometry. And so this is something that we're also just working on completing, and that we hope to publish in a preprint also in the coming weeks. And then finally, and that's for today, we have Rosalie's works, which is on measuring the inner and outer proteins in EVs. And I think we're all familiar with measuring proteins on the outside of EVs, but often when you want to measure them inside, we need to lyse all the EV and we lose this co-expression information. And so this is what Rosalie has been working on. And as I was saying before, technology development can take many years. Rosalie started in our lab as a master's student many years ago. And she had actually early promising results, but then every time we wanted to demonstrate it at a higher level, then things didn't work, and then she had to go back to the drawing board. And so there were quite a few iterations of that, but we're very pleased that she kept at it with grit and a lot of determination, and she was able to bring this work to fruition and analyze proteins both inside and outside in a highly multiplexed manner. And so we're very grateful for Rosalie for all her efforts, and I'm looking forward to her presentation. And so Rosalie, I'll without further ado, I'll let you give you the floor here.
As David said, today I'll present our work on the EV antibody microarray for multiplex inner and outer protein analysis. We looked at many studies that studied EVs, and as you probably know, EVs are central to intercellular communication, and there have been many studies into their roles in cancer pathogenesis and other diseases. And what we know from all those studies is that those functional effects are linked to their biomolecular cargo. And EVs are actually quite complex. They have like a complex cargo of different types of molecules, which include mRNA, microRNAs, DNAs, lipids, and also what will be the focus of today's presentation, proteins. So proteins can be divided into two broad categories. So we have cytosolic or inner proteins, which include cytosolic proteins, but also inner leaflet portions of proteins. And then you have outer or surface or membrane proteins, which include outer leaflet protein domains, as well as other surface presenting or adsorbed proteins. So given the enthusiasm around the biological and pathological relevance of EV cargo, there have been a lot of methods developed or adapted in recent years to study EV proteins. So these include mass spectrometry, flow cytometry, microfluidic and integrated platforms, affinity binders and affinity binder arrays, such as antibody microarrays and atomer arrays, and many others. For a lot of these methods, the default is to look at outer EV proteins, and looking at inner proteins presents additional challenges. And these challenges are worth addressing, as many key protein markers are in fact inner proteins, from common EV proteins like shock proteins and ALEX to more disease-relevant proteins such as mutated actors in cancer. And there are ways that exist to look at such inner cargo, albeit with some limitations. So for example, one can lyse EVs prior to analysis and get some population averages on inner protein expression. Or you can first capture your EVs by targeting an outer protein marker, then lyse it so you can correlate the inner protein reading with the surface marker expression. And one other possibility is to use surfactants in solution to make EVs more porous. But then you run the risk of inner proteins diffusing out, so you don't get as much sensitivity as you could otherwise. So knowing that, how would one improve inner protein retention and maximize co-expression information? That's the problem we embarked upon. So when it comes to co-expression analysis, antibody microarrays are an attractive solution. So it was pioneered by Jorgensen and colleagues in a 2013 JS publication. And antibody microarray offer high throughput combinatorial EV protein analysis by making it possible to capture many EV subpopulations based on surface expression, then perform protein detection on those EVs using fluorescence detection. And our group actually worked with antibody microarrays in the past for protein analysis, so it was a natural starting point. As for inner protein detection, we looked through cell immunohistochemistry methods for guidance, where we learned about a process that's called antigen retrieval, or AR for short. In immunohistochemistry, AR is a method used on formalin-fixed paraffin-embedded samples to recover antigen sites hidden by fixation. It uses a treatment, usually heat-based or enzymatic, often combined with acidic or basic buffers, to perform hydrolytic breakage of inter and intramolecular crosslinks and protein renaturation. So what we decided to do is to explore how those ideas could be applied to EVs to avoid inner protein diffusion out of the EV, as well as using fixation to preserve antigens within the EV. So the main difficulties with inner protein detection are that there are a lot of those proteins that are soluble and they are constrained within a lipid bilayer. So if you have the lipid bilayer, like it follows that if you add antibodies, they can't directly detect inner proteins unless you do something about it. So the main challenges here is to balance antigen retention, which is generally achieved through fixation, and antigen availability, which is reduced by fixation, but can be restored using some form of antigen retrieval. So fixation is what prevents proteins from diffusing out after we permeabilize the EV. But as you can see, it also creates this network of crosslinks within the EV, which reduces the probability of antibodies actually finding their target. So to alleviate this, we can use AR to reduce the number of crosslinks between proteins without removing all of them, because we still want to preserve antigens within the EV. And we do this through hydrolytic breakage, making it easier for antibodies to get through and detect their target. So combining these ideas, we developed the inner and outer extracellular vesicle protein analysis platform, or EVPIO for short, of which we have an overview here. So it consists in a glass slide with 16 10 by 10 antibody microarrays per slide that are inkjet printed on an aldehyde functionalized surface. Replicates are organized in rows, and the slide is superimposed with a slide gasket, a well gasket, to allow up to 3 different detection antibodies to be incubated at the same time per array, which are then imaged on separate fluorescence channels. So here is the general EVPIO workflow. So as I mentioned, we spot capture antibodies on the slide to form our microarrays. Then we block the surface with BSA and other blocking agents. Then we capture EVs on the surface overnight, fix them using formaldehyde, and then perform an AR treatment. I'll get back to that after for the details. Then we quench the surface using BSA and glycine, incubate with detection antibodies. So these are barcoded. Again, I'll talk a bit more about that later. And then we incubate them with oligo trees of up to 3 different fluorescent colors and perform detection using a microarray scanner. For AR optimization, we used EVs from the A431 epidermoid carcinoma cell line, which is a well-known proteomic composition. And we purified these EVs using size exclusion chromatography and obtained a population that was around 70 to 300 nanometers big, with a peak around 120 nanometers. And as you can see on our TM image here, they have the classic cup shape that's expected for EVs. So to perform the AR optimization, we captured EVs based on their CD63 content before performing the AR treatment. And we detected 2 different proteins, so HSP90 as a model inner protein and EGFR as an outer protein. And what we initially did is we tried using existing cell protocols, which suggested times on the order of 10 to 20 minutes. And that ended up being way too harsh. So we adjusted our parameter ranges according to that. So we looked at 4 different parameters. We looked at the buffers that we could use. So we had a buffer containing urea, TBS, and PBS. We looked at 3 different temperatures. So most cell protocols use very high temperatures, but since this is quite harsh, we also looked at 50 degrees Celsius and 70 degrees Celsius. And we also looked at different times, so 1 or 2 minutes. So you can compare that to the 10 to 20 minutes that we tried initially, and you can see how different it is for EVs. So the data that you can see here is actually using 0.05% Tridonex 100 as a pre-treatment for permeabilization. But actually what we found is that this actually wasn't needed and that we obtained better signals if we skip that step altogether. So the data here is for without any prior permeatization. So just the buffer with at that specific temperature and that specific time. So that way we can see that our array captured EVs are actually different from the formalin specs paraffin embedded cells in immunohistochemistry. So for which existing protocols are optimized. So they're much smaller, they're more fragile, and they're also easily permeabilized due to the absence of supporting outer structures, which include normally like extracellular matrix, cell junction, cytoskeleton, etc. So it was interesting to see how different it was when applied to EV. So the chosen condition that we use going forward is this condition of 1 minute in 90 degrees Celsius PBS, which was found to be optimal for the detection of HSP90 and EGFR in CD63 positive EVs. So here you can see like an example of what the array looks like. You have the negative capture controls that are indicated as well as our CD63 spots. And down here you have quantification of that data, which shows a pretty high robust signal for HSP90 for that condition. We tested AR further using the plus detection for 2 different duos of proteins, which include 2 additional inner targets for validation, so Alex and HSP70, and integrin beta-1 subunit as an outer target. This used a combination of species detection and biotin strip detection for multiplexing. And you can see what the arrays look like here. And this is the quantification of the data. And this confirmed robust signals for all proteins tested in several subpopulations, not all of them, because it depends on the surface expression of the EVs of that particular protein, the capture targets on the x-axis. But we see a lot of consistent signals that are on the order of like 10,000 RFUs. So we were pretty happy with that. So we tested a different construct for branched DNA signal amplification. So since inner protein detection can actually be challenging, especially when you have lower abundance, we included signal amplification on the platform to ensure that we could get good sensitivity. So we tested different types of constructs for that. Here are illustrated a linear construct, a 2-branch construct, and a 4-branch construct with increasing numbers of fluorophores. The simplest design involves a 15-mer unique orthogonal barcode for target detection, an imaging strand, and a fluorophore strand for labeling. And larger constructs have additional, what we call bridge strands, with intermediate barcodes to form branches and hybridize more fluorophore strands. Those additional unique barcodes are designed to minimize cross-reactivity within the strand. And there are many great things about this kind of signal amplification, which is why we chose it. It's highly customizable, it's compatible with multiplexing, and trees can be assembled in parallel to the assay. They require some thermal cycling to ensure proper assembly, but you don't have to do this during the actual assay. You can do it in parallel so that it doesn't increase the experiment time. And prior to this, we tried a bunch of different ways to amplify signals. We tried RCA, and like that one was very long, was also costly because of the use of enzymes and was also temperature sensitive. So like this solution is simple and pretty straightforward, so we were happy with that. So this is just a snapshot of the combinations we explored to actually optimize our assays for oligo-based labeling and detection. So we had already optimized buffers for use with antibodies, and we had to go back to the drawing board when we introduced oligos because they bind to the surface in a different way, and like they can introduce background of their own. So we optimized for oligo binding as well as the detachment of the oligo and to reduce the background as much as possible. So we explored additives such as sheared salmon sperm DNA and dextrin sulfate that are used for blocking and encourage binding respectively. So I won't go into the data, like all the information is available in the supplementary information of our publication, but just to give you an idea of like all that was explored to optimize the assay. So this is the result of our DNA signal amplification optimization. So for this, we used CD9 detection to compare the construct against standard detection using a biotin label and a fluorescence tryptophan probe. And you have a variety of capture targets. So we used the canonical EV tetraspanins as well as a cancer marker because here we use SW43 EVs, which are cancer EVs. So this is like a cancer-associated marker. So we found using this that the two-branch tree performed comparably to the biotin-tryptophan benchmark. So the two-branch tree signal SNR is in yellow as in compared to the biotin-tryptophan signal in blue. In the meantime, the four-branch and linear construct gave lower SNR than the benchmark. And one aspect to consider is that brighter trees with more flow force are also bigger. So that could mean that some steric hindrance is happening if there are a lot of binding events, or even if the construct have to bind in a crowded environment. It's possible to use the expected target abundance to guide the choice of tree. For example, to use a bigger tree for a less abundant surface target and a smaller tree for a higher abundance of inner target. But for simplicity's sake, we focused on the two-branch tree based on this data. So after that, once we had introduced signal amplification, we introduced multiplexing to the assay gradually. We first tested two different duplexes of one inner and one outer target in HC29 and W403 colorectal cancer EVs. Before moving on to triplex detection as shown here. So here I'm only showing the HC29 data. We also have in the paper the data for the SW43 EVs. We tested all three construct types. So we found that we could consistently detect inner target HSP70 above background with two-branch trees, as you can see in red here. And we got overall higher signals and successful multiplexing with a two-branch tree, which helped validate its choice as the main signal amplification construct for future AV-PIO assays. We also went ahead and compared the signal we got in multiplex assays compared to assays performed in singleplex and calculated the ratio between the two. So encouragingly, we found that that ratio is equal or superior to 1 in the majority of cases. So in the x-axis here, you have like results for different targets and different constructs for signal amplification. So we got an average ratio of 2.9 and a median of 1.4. So we can say with confidence that overall, in like most cases, we don't get any signal loss with multiplexing. So after that, we moved on to doing some more phenotyping of our colorectal cancer cell line EVs as a proof of concept. So you can see here what the experimental design looked like. We used arrays that are targeting the canonical EV tetraspiden CD63, CD9, and CD81, as well as a CAM, which I mentioned before, which is an adhesion molecule overexpressed in epithelial cancers. As for detection markers, we chose 12 proteins based on their expression in EVs, their biological function, or their involvement in cancer. So we had the same tetraspiden used for captures in blue here, as well as a few integrin subunits known to be involved in organotropic metastasis in green, and some common EV proteins like EAT-shock protein and ALEX, as well as Claudin-2, a type junction protein relevant to colorectal cancer. And that one is detected based on its inner leaflet domain. So all the inner proteins are in red here. The schematic shows how proteins to be detected were arranged in triplexes for multiplex detection. We matched each protein to a fluorescence channel based on this expected expression. For example, we expected high tetraspiden abundance. So we put them in our 488 channel, which with our imaging setup is actually the channel with the smallest bandwidth, and it's a bit less sensitive. We also used CFDA stained EVs as capture control and added a positive detection control with direct spot detection using the conjugated antibodies and trees. So here you have some quantified data of our phenotyping results. So some phenotyping highlights here. We had a high signal in CD9 plus EVs in both cell lines. And we also found that integrin alpha 6 had the strongest detection signal. So this is in green here of all of the targets we looked at. We also, We also found that ALEX was consistently detected above threshold. So in red here, so it was our strongest inner protein that we detected. Of note, we got substantial unspecific binding that occurred on our anti-GFP negative control spots, which led to higher variability and higher cutoff values. And this is especially marked in the Site3 channel. So you can see the green line of the cutoff here. And again, that comes down to our imaging setup. So because of its larger bandwidth and the proximity of its peak to the autofluorescence peak, then we got a bit higher background with this. So the imaging setup is something very important to consider when you're running assays like these. So here we got the same data presented as heat maps, and the way it's presented here is to accentuate differences between the two cell lines. So you have the RFUs for HC29 illustrated here as is, and then you have the ratio of the RFUs for SW43 and HC29 EVs to highlight the differences. So several things we took from this. First, that in the EPCAM plus subpopulation of SW43 EVs, we got a lot of differential expression compared to HC29. So higher signal for CD81, CD9 integrin alpha 6, up to 10 times higher, and also for inner proteins HSP90 and ALEX up to 6 times higher. And we also got notably lower expression of CD82. In CD63 plus SW43 EVs, we got lower integrin beta 1 expression. And in CD9 plus and CD81 plus SW43 EVs, we got higher signals for CD81, integrin alpha 6, and Claudin 2. So yeah. We also ran the experiment on another biological replicate and looked at how the values correlated together to make sure it was reproducible. And we got high coefficients of determination for both cell lines tested, which indicated good reproducibility of our assays. Finally, we also compared the signal we obtained with and without AR for our different targets to assess what the process did in terms of signal. So we computed the AR to no AR ratio and got ratios mostly higher than 1 for inner proteins, which is what you wanted. You can see it here, for example, for HSP70 and for Claudin 2 and HSP70, higher signals, like much more signal in SW43 EVs. And for other proteins, though, there's some loss of signal occurring. So it's a trade-off. Since other proteins are already more accessible, they probably get more hydrolytic breakage from the get-go than inner ones. So you have to balance how much AR to do depending on the location of the protein too. From this, we concluded that it's better to use AR only on inner targets unless you want to do some further optimization for a specific subset of proteins. So going forward, doing separate slides for AR targets versus non-AR targets could be a good way to take advantage of this. So we have several points that we can discuss for this. So one big aspect of our assay that's very advantageous is that it offers a lot of antibody and for flexibility. So since the antibodies are conjugated in-house and the trees are also built in-house, including some barcoding, you have a lot of integration possibilities. You can use any antibody from any species, any fluorescent channel, so you can mix and match according to target abundance. So it gives you a lot of flexibility in that regard. Regarding the loss of other proteins upon AR, other protein signal upon AR, so when we optimized this, we performed the optimization in a sequential manner. So some things were added later based on what we found as we developed the assay. So there would be space to go back and perform a more systematic, exhaustive optimization, try to get more broadly available, applicable parameters, or alternatively, if you really want to look at a specific small subset of targets, you can optimize for that using similar parameters to those we use. But like a straightforward solution is, like I mentioned, to consider separate assays for inner and outer proteins. And the customizable amplification trees have a lot of advantage in this assay. So they're assembled in parallel. They're used in mild conditions, so you don't need to use heat or anything like that on the assay that could affect your protein sensitivity detection. And you can also select them based on target abundance, as I mentioned, with the different tree sizes. Regarding background and threshold, so one thing to consider is the careful selection of flow forwards and imaging setup. So what the bandwidth of your fluorescent channel, for example, can have a big impact on your background signal. And a wise way to go about this is to match the channels you have to target abundance to make sure that lower abundance proteins can be detected. In terms of quantification, the data I showed you allows direct comparison across samples. So I can compare the same co-expression item across HE29 and SW403 EVs, but it's not as straightforward to compare co-expression items within an EV, and that's because we use different antibodies with different binding properties and also different flow pores. So there could be some normalization that could be developed for this. But it's something to consider that you can only get semi-quantitative information within a given sample. And finally, we believe that using AR combined with high resolution studies like TEM studies, we could look at how it affects EV integrity, and that could inform future single EV studies that would want to look at inner EV proteins. So based on the EV-PIO assay, we have ongoing work that is meant to look into applications of the assays for clinical samples. So we're currently optimizing the assay for its compatibility with plasma samples. So using, for example, spike-ins as well as pooled blood samples on the assay. And we're also optimizing the sample processing pipeline. So it's going to be processed probably differently from how you would go for cell line EVs. So we're looking into that and how to make it all work. And the goal with this is to go forward with the analysis of colorectal cancer patient samples. So we plan to have test and training cohorts of patient plasma, and starting from a 20+ protein panel to look at co-expression patterns with the goal ultimately to model for classification of patient samples. With this, I'd like to thank the whole team at Juncker Lab, especially my colleagues who also work on our different EV projects. So that includes co-authors Professor David Juncker, Molly Shen, Philippe de Corin-Martin, and Lorena Oliveira, as well as other EV working colleagues. So I thank you for your attention. And here's a quick summary, take-home message from my presentation. So EV-PIO platform offers flexible, high-throughput multiplex analysis of inner and outer EV proteins with high antigen retention, customizable signal amplification, and compatibility with standard lab equipment such as a microarray scanner or a fluorescence microscope. And it has potential for extensive combinatorial protein profiles to study various conditions. So we gave you an overview with our proof of concept phenotyping in comparison of HE29 and SW403 colorectal cancer EVs, but we plan to show it even more with ongoing work on colorectal cancer patient samples. So thank you for your attention.
Thank you, Rosalie. That was a great presentation. And my friends, it is time for the discussion. So first of all, I noticed that there has already been some discussion in the chat box, and Professor Juncker has been answering some of the questions that have come in. But we will go through these anyway and just make sure that all of the answers are there. So let's start out with Schiatroma Cataldo, who is asking, I'm not sure, maybe the microphone is not working, but asking what the fixation protocol was.
Oh, for the assay, it's 4% PFA for 10 minutes.
That's right. So 4% PFA. And for how many minutes?
10.
Okay, very good. So now Heather Tanner.
Thank you for your presentation, Rosalie. It was really interesting. I just was curious which type of antigen retrieval you adapted. Was it I think I was reading there's only two, protease induced or heat induced?
It's heat induced. Yes, it's an adapted version of heat induced using a neutral buffer instead of a pH basic or acidic buffer.
And you couldn't reuse the same sample in the multiplex as the antigen retrieval. You had to just use like aliquot from the same sample for each one. So
sorry, I'm not sure I understand the question.
I don't think you can't, after you do AR on one sample, you can't use that sample again. You need to take an aliquot from the original sample, or are you able to use the exact same sample you used?
Oh, so we performed AR directly on the slide once the EVs are captured. So we performed the detection, the multiplex detection directly after on the same slide is the same sample that was AR.
Oh, okay. Thank you.
No problem.
Yeah,
Go
thanks
ahead.
for the question. If I may just, I think the AR protocol, I mean, we tried a lot of more complicated things and chemicals, but in the end, it's a 1 minute at 90 degree on a slide. Right? So you have a heated plate, hot plate, you put your slide, or maybe, no, you immerse your slide in this 90-degree solution for 1 minute. And so it's actually very fast and fairly straightforward. Maybe Rosalie, you can comment on that.
Yeah, it looks like that. It literally looks like that. We have like a beaker with PDS in it. We heated up to 90 degrees and we dunk the slide in this for a minute. Yeah, after that, we rinse it to remove extra salt, and then we put the gasket again and continue the assay. Yeah, so it's directly on the slide.
Very clear. Another, maybe a follow-up question on retrieval, Joshua Spitzberg.
All right, thank you. So I was wondering, I think you alluded to this, how much do the optimal retrieval parameters change, the temperature and time across, if you did 10 different proteins? I think you showed 2 in one graph, but I'm wondering really how much does this, if you've explored this?
We didn't explore it per se, but we saw the effect of this when we moved from like those 3, like you remember we did the one for the optimization and then validation with 2 other ones, and then we moved on to a larger panel. So when we moved into the larger panel, we saw some variation for sure, because some of the signal was not increasing all that much with AR, which is what we showed in the violin plots at the end. So it's likely that with a larger array of targets, it could vary more. It depends on like how the protein is interacting with other proteins inside the EVs, like how buried it is, how linked it is. So different proteins will need to be broken hydrolytically in a bit differently. So that's something to keep in mind. Like if we're going to do larger panels, we might look into adjusting it a little bit.
I was just wondering, did you come across any that, for example, seemed incompatible?
There are some proteins we didn't get any signal for, so that could be incompatibility, or that could be like undetectability below threshold, but that's hard to say.
So if I may just add, so we started the process with HSP90 as a model, but then when we found out it might not be exactly this, or the condition we find might not be the optimal condition for all proteins, and so there's already a lot of iteration here, but I think this is a demonstration that this is really possible. But I think there is certainly room for improvement. There can be like systematic, like a design of experiments or Taguchi methods that could, for example, be used for optimization while also targeting a larger set of proteins that what we have done so far. And so I think this is a starting point, and I hope that others will take this as a starting point and develop it further.
So yeah, I see there's a question about biofluids here, and like what I can say is yes, it's much more complicated. Yes, we had issues. We're working on it, and we've got promising results so far. So we had to change the way the samples processed, and we had to review a little bit the buffers, but it's adaptable. Like SCC plasma, the issue we had was not like noisiness per se, but like loss of signal because it got too diluted. So we were looking at an approach that's a bit simpler and bare bones, using filtration of the plasma sample and like a bit of centrifugation to remove debris, but like not too much so we don't dilute things overly. So yes, there are a lot of things that can come in the way of the assay in biological fluids.
Yvonne, did you have a follow-up on that?
I didn't. Thanks ever so much, Rosalie, for that. That was a great answer. What is your starting sample volume for plasma? The issue that I'm having is that I'm working with mice. So my starting sample volumes for plasma are absolutely minuscule. And so once I've sect them as well, it becomes a little bit like homeopathy. And I'm wondering whether all I'm seeing is noise and not signal, and whether that's because of, like you mentioned, sort of steric hindrance and stoichiometry of either protein corona or contaminating lipoproteins, or I have no idea what's going on, but it seems like it's much harder to do all of this stuff with plasma than it is to do with neat cell culture samples.
Yeah, it's definitely harder. Regarding like sample volumes and the use of SEC, so one thing that we found that works pretty well is like to dilute the plasma sample 1 in 3 and then just perform a syringe or a centrifuge filtration with it. So there will still be like proteins in suspension, but it's going to remove like a lot of the cell debris. But if you're lucky, the remaining proteins in suspension can even help like block the surface, like in our case. So that we got higher SNRs, like we compared with SEC and we got higher SNRs doing this for example, canonical EV tetraspin.
Okay, that's perfect. Thank you.
Yeah, so yeah, I think from my experience, we started like SEC, like with cell supernatant, but that really didn't work. And then in the end, we simplified the protocol and have just a filtration step, but actually with fairly large pore filtration, and right, with I think 400 nanometer. So to try to retain more, and I mean, I remember this discussion also came up, I think, in previous EV clubs here. And so for us, it also, and also part because we have the antibodies, of course, that serve for the purification on one hand, and also for the recognition. So here we can tolerate maybe a bit more contaminants than you might maybe with some other methods that don't use antibodies. But then so using a fairly large filtration to retain as many EVs as possible, and then a centrifugation step, that sort of gave us the best result on this microarray, both for outer and for inner proteins in this case.
Hello, Rosalie. Thanks a lot for the talk and for the previous answer. I have another question about fixation, and I was just wondering if your retrieval protocols might affect the bond that you create between the antibodies and your surface. If I remember, one of your first slides showed that you use aldehyde.
So the way we actually tested for like signal loss is like empirically. We looked at how much of the signal we retained. And like in early experiments, if you recall, when we used EGFR as an outside marker, we retained a lot of signal even with the AR process. So that's how we deduced that detachment must be like minimal in this case, since we retained our detection signal.
Thank you.
Very good. Thanks for the questions. And let's go back to Joshua Spitzberg. Joshua, you had a few more questions here, so please go ahead. And
I think one of them had been answered already. I was asking, what are the conditions for comparing AR to no AR? Was it AR, was the denominator like PFA fixation? And I think somebody had already asked and then it had been answered.
Both are fixed, yes.
But the other question I had was going back to steric hindrance. I was wondering, have you seen an upper limit on how many antibody oligo constructs you could profile? I think you went up to 6 or something. The slide was a little quick, but then I saw, future work of prospective panel of 20. So I was wondering, can you do 20, on the same sample of EV, or, is there an upper limit on, one sample, and then you're going to do 20 across, some other, larger number of samples?
So steric hindrance, yes, it's a concern for sure. I showed this that up to 3-plex, we didn't see issues with like signal reducing because we had many at the same time. And honestly, the 20-plex, the plan is not to throw all 20 at the same time, is to perform 3-plexes like this, but like up to like 3 at a time for 20 total.
Could you maybe just walk us through the slide, Rosalie? I think it might be a little bit confusing.
Yeah,
I'm curious, can you go beyond, have you tried going beyond 3?
I didn't try, I could. The reason I didn't try is because the microarray scanner we use has 3 channels. So I would have had to move imaging setups to try more. But I think it's definitely possible. But personally, like I believe like my educated guess is there will be an upper limit of like targets at the same time, like depending on their respective expression, where you're going to get too many trees and you're going to start to lose signals on some.
I mean, we did with the single EV analyzers, we're also looking at multiplexing, and we looked at up to 20 without DNA, without the trees attached to them per se. But so there we could do 20-plex, and we didn't see a difference with single plex. So it seems like you could detect many proteins on single EVs. I mean, if you have big trees, that might be, and you all attach them simultaneously, that might be an issue. But from the antibodies alone, it seems like multiplexing is a possibility. Now, with the microarray approach here, we have different wells, and in each well, you have an array of capture antibodies, and you have up to 3 detection antibodies, which are dictated by the 3 fluorescent colors of the microarray scanner. So when we have this approach, so here we have the multiplexing by having different wells with different samples. So I mean we can't really explore the multiplexing ability in this kind of configuration, but we can get multiplexing by having these replicate kind of spots and replicate samples as well, right? That means, so what's the volume of one well you need in sample? I think that was kind of interest to some people, and then, well, how many more wells you have, how much more sample you need.
It's 50 microliters per well that we use. So depending, like for EV from cell lines, we didn't dilute it, but for plasma, we dilute 1 in 3. So like you actually need like about 20 microliter per well, that means.
Yeah, and thanks to Molly, Molly Shen had put an answer here too in the chat box. That was, yeah, pretty much what you just said, Rosalie, that even as little as 10 microliters of pure plasma might be needed depending on the dilution. So yes. That's good to hear. Good. All right. Let's see who is next here. Erez Item. So Erez has asked, when you compared single and multiplex, was it just for detecting antibodies or also for capturing antibodies? Then furthermore, do you have any ideas on developing a standard curve or some kind of a QC measurement for the assay?
So regarding the multiplexing, it was just for detection. The capturing part was the same. So it was just like using the different antibodies in separate wells or using them 3 at a time in a well and comparing the signal. Regarding standard curve or QC, we thought about this several times, but it's actually, I would think you would have to do some kind of like EV dilution and have a measurement for each co-expression item on the different concentrations of EVs, and then you could try to draw a curve that would be specific to that captured subpopulation and that detection target. You can see how it could be very time intensive if you do this across like a very big panel, but that's definitely a possibility. Like you could select some co-expression items and do this and have an idea, but if you want to quantify everything, it's going to be a lot of work.
Can you hear me now?
We can. Go ahead.
So first of all, thank you for the answer. And I know that generating a standard curve is not an easy task. And I think that's something that a lot of us developing those essays facing. So I was wondering how you think about it. But I think that's the combination of multiplexing in both the capture and the detection is actually very interesting and can really increase the results. So thank you for a great presentation.
Thank you.
Thanks, Erez. I believe that the next question may have been answered. This is from Patricia, who asked, what kind of heat did you use for AR? So you very clearly showed the illustration there. But Patricia, feel free to jump in. I mean, did you try other kinds? So Patricia mentions here microwave, heat vapor. You think that there could be another way to do it?
So we thought about using enzymes and played with the idea for a bit, but it was a whole other optimization area, so we didn't actually delve into it much. We used this because it was like the most accessible, and microwave, it was hard to control. Like if you compare it to using buffer like this, where you can control the temperature, like you cannot really easily control your microwave or your pressure cooker. So that's why we went with this, because it's more modular.
Mm-hmm.
And vapor, did you look at vapor?
No, I didn't look at
vapor. Huh? Yeah.
No, we didn't. We thought about some other options, but not vapor. Yeah.
Or direct heat, we also didn't. I suppose that means infrared, or I don't know.
Dry
that
heat.
Would be the other one.
Yeah.
Yeah, or dry heat in an oven, but no, we didn't look at that either. So I mean, this might work. I don't know.
Well,
So
usually
like
you require a buffer.
Control. Yeah. Good. All right. Thanks for the question. And then it looks like we have one more question, and this is from Elizabeth Delar. Please go ahead.
Hi. I was just wondering, when you did your antigen retrieval optimization, you used the HSV90 and EGFR, and given that they've been shown to be abundant in supermare populations, I just wondered whether the differences you see between the conditions may be to do with sort of whether those are kind of associating with the vesicle surface in some conditions and disrupting in others, and sort of how that might be influencing what you've seen.
So we performed some validation on the EV samples to try to make sure that there were actually EVs in there. we cannot like be 100% sure we don't have like super mirrors or other conditions in there, or the effect of that.
Your negative, sorry, your negative control or your controls at low, there's conditions where you have no signal, and I think your PBS control also shows no signal, right?
Yes, yes.
So that's maybe one validation. And I think when we started this, I'm not sure how much was known about super mirrors, so I don't think we
Yeah,
I
actually,
think
I was telling myself that too.
So,
these experiments are a few years old, yes, so I'm not sure how familiar with the concept of super mirror we were back then. But yeah, that's definitely something to think about.
Thanks.
Excellent. Well, we have come to the end of the questions that are in the chat box. We do have one more comment from Molly Shen here. Molly, did you want to just go ahead and share this? Because I know that the people who are watching this on YouTube won't be able to see the chat box.
Sure. I just wanted to, I guess it was also mentioned in some questions. While Rosalie presented our array in 16 wall format, there's actually a lot of flexibility in how we fabricate the array. So routinely we do both 16 wall, which only needs 50 microliter of the sample, as well as the 64 wall format, which only needs 20 microliter. We do have the possibility of going even lower volume. So if any of you have any need in terms of running assays with low volume, we do think that microarray could be a good alternative for you.
Sounds good. Thanks for the clarification, Molly. All right. Well, thank you all for joining today. It's been a very interesting discussion, and I've learned a lot about the array and about what can be done with it. And special thanks, of course, to Rosalie and to Professor Juncker and to the whole Juncker lab. Thanks so much. We wish you the very best as you continue this exciting work. So thanks again. And we look forward to seeing you on a future EV club. We still have a few slots that are open in February. So if anybody has something hot that they want to share, please reach out to me and I'd be happy to get you on the schedule. All right. Well, thanks everybody. And remember those abstracts for Friday night.
Thank you.
Thanks all for all the great questions.
Take care now.
Thank you.
Take care. Bye.